All right, please. Please be seated.
Hereby, I open this academic ceremony in which Hongyu Zhao will defend the academic thesis Restoring Lost Memories, PDE4 Inhibition and Subliminal Reactivation as Novel Approaches to Counteract Sleep Deprivation-Induced Memory Deficits. May I invite you to present a summary of your study and the conclusions of your thesis?
Dear Proctor, dear members of the Defense Committee, dear supervisors, colleagues, friends, and family, and audience at home, thank you very much for coming to my defense for the thesis titled Restoring Lost Memories, PDE4 Inhibition and Subliminal Reactivation as Novel Approaches to Counteract Sleep Deprivation-Induced Memory Deficits. After a night of bad sleep, you feel exhausted, your thinking slows down, and hard to remember things. Sleep deprivation can affect different groups of people, such as healthcare workers, like doctors and nurses, academic researchers, and students. Sleep deprivation can lead to various health issues, such as cardiovascular health risks, emotional dysregulation, cognitive decline, neurodegenerative risks, and immune suppression. Notably, it impairs hippocampal memory, which is the focus of my thesis. Our previous animal studies have already been shown that hippocampal memory after sleep deprivation can be restored by non-selective PDE4 inhibitor and object reactivation. Based on those results, my PhD focused on two key directions. The first, while non-selective PDE4 inhibitors showed effect, the specific subtype remained unclear. So we decided to test which selective PDE4 inhibitor is responsible for this effect in animal studies. Second, we already know optogenetic reactivation is an useful method, but we cannot use this method in humans. So we tested an alternative approach, that is subliminal reactivation in humans. The aim of my thesis is to find ways to restore memory deficits caused by sleep deprivation. The first, test the function of selective PDE4 inhibitor in animals covered in Chapter 2 and 3. Second, test whether subliminal reactivation can bring back lost memories in humans covered in Chapter 4, 5, and 6. Let's start by animal studies. This slide shows intracellular mechanism of non-selective PDE4 inhibitor, specifically through CAMP signaling pathway. In the hippocampal neuron, CAMP activates PKA, PDE4 block CAMP. During sleep deprivation, PDE4 protein level goes up. It decreases CAMP signaling pathway, which in turn leads to poor memory. PDE4 inhibitors help by blocking PDE4 levels. It increases CAMP signaling pathway, which in turn leads to good memory. After understanding the mechanism of non-selective PDE4 inhibitor, the next question is, which specific PDE4 subtypes are involved in my thesis? There are 4 PDE4 subtypes, A, B, C, and D. PDE4A, there is no selective inhibitor available for this. PDE4C is not expressed in the brain. PDE4B is assumed to be more related to affect, such as depression and anxiety. PDE4D is assumed to be more related to cognition, such as memory enhancement. My thesis focused on PDE4B and D. How did we explore these animals? Let's have a look at animal experiment design. Mice first complete learning trail, including pattern separation and object location memory. Then 24 hours later, they complete test trail to check whether they still remember the stuff in the learning trail. Specifically, in pattern separation test, mice were sleep deprived for 6 hours before the test trail to disrupt memory retrieval. In the object location memory, mice were sleep deprived for 6 hours after the learning trail to disrupt memory consolidation. During sleep deprivation, we give them vehicle PDE4B or D inhibitor to check whether these inhibitors can counteract sleep deprivation-induced memory deficits. This is the result of animal study. Since pattern separation and object location memory tests showed similar results, we show pattern separation test as an example. On the y-axis, we have memory performance. The higher the value, the better the memory. In the non-sleep deprivation group, in the vehicle condition, this is the baseline of memory performance. However, after sleep deprivation, memory performance dropped down in the vehicle condition. Interestingly, PDE4B or D inhibitor can improve memory performance after sleep deprivation. It's worth noting that PDE4B can impair memory performance in the non-sleep deprivation group. After finishing the animal studies, we move on to the human studies. The aim of human study is to answer an important question. What happens to memory after sleep deprivation? Are they gone because they weren't stored? That is availability issue. Or are they still there but stored suboptimally? That is accessibility issue. To answer this question, as we mentioned earlier, we used a method that serves as a human applicable version of optogenetic reactivation, that is subliminal reactivation. So what is subliminal reactivation? This is very brief stimuli without conscious awareness. I will show you an example. In this demo, you will see a word flashed very briefly, followed by a mask similar to a QR code. Here it comes, get ready, because it will be very fast. 3, 2, 1. Did you see anything? Probably not. So that's exactly the point. We tested whether subliminal reactivation can bring back lost memories without conscious awareness. Then we used subliminal reactivation in human studies. In Chapter 4, we tested optimal subliminal duration to use in the following experiments. In Chapter 5, we recorded EEG during subliminal reactivation to collect brain markers to validate method. In Chapter 6, we check whether subliminal reactivation can restore memory after sleep deprivation. Let's have a look at EEG experimental design. Participants first complete learning trail, including words and pictures. Then 24 hours later, they complete test trail to check whether they still remember the items in the learning trail. Before the test trail, we'll give them subliminal reactivation. Specifically, either subliminally flashed learned items or mask-only condition. We recorded EEG during both subliminal reactivation and test trail. This is the results of EEG study. We wanted to understand how subliminal reactivation are processed in the brain. Due to time limitation, I will show you verbal learning test as an example. During subliminal reactivation, we recorded N200 in the parietal and occipital region. The red line represents old stimuli. The blue line represents new stimuli. The black line represents only masked stimuli. We found new stimuli triggered a more negative N100 than old stimuli than only masked stimuli. This suggests that processing new stimuli require more cognitive efforts than old stimuli than only masked stimuli. In the test trial, we recorded P100 and N170 in the parietal and occipital region. The solid line represents old atoms with subliminal reactivation. The dotted line means old atoms without subliminal reactivation. We found subliminal reactivation triggered a more positive P100 and less negative N170. It suggests that enhanced P100 suggests subliminal reactivation accelerated sensory processing. The reduced N170 suggests that subliminal reactivation accelerated discrimination during memory recognition. Importantly, all these EEG results were supported by behavior data. We found that subliminal reactivation can improve memory performance compared with no subliminal reactivation. This suggests that subliminal reactivation can be processed in the brain and can improve memory performance. After validating this method, we move on to the main question: whether subliminal reactivation can counteract sleep deprivation-induced memory deficits. Let's have a look at EEG study experiment design. This is based on the EEG study experiment design, but introduced sleep deprivation on the night of the learning trial, including control, half-night, and full-night sleep deprivation. Similar to EEG study, participants first completed a learning trial, including words, locations, and objects. Then 24 hours later, they complete test trial. Before the test trial, we give them subliminal reactivation. This is the results of sleep study. On the y-axis, we have memory performance. The higher the value, the better the memory. We found significant effects in the verbal learning test and object pattern separation test. In a no subliminal condition, we found both half and full night sleep deprivation impairs memory performance compared with control group. However, after subliminal reactivation, memory performance improved across all groups, including control, half-night, and full-night sleep deprivation. Interestingly, both half and full-night sleep deprivation return to the same level as control group. This suggests that subliminal reactivation can improve memory in people who have sleep deprived and with normal sleep pattern. In conclusion, in animal studies, we found PDE4B or D inhibitor prevents sleep deprivation-induced memory deficits. And since we already know PDE4A can also improve memory performance, our findings suggest that non-selective PDE4 inhibitor may be more effective than just targeting only one subtype. In human studies, we have two key findings. The first, proof of concept: subliminal reactivation can revive memory in humans. Second, proof of principle: the memory is not fully lost, but hard to retrieve. This answers the question: what happens to memory after sleep deprivation? They are still there, but hard to retrieve. That is an accessibility issue. So with that, I would like to thank our sponsors, the MES, the participants, my supervisors, and now I hand over back to Proctor.
Thank you very much for your very clear presentation. The opposition will now be opened by Professor Sambet, Professor of Educational Neuroscience at the Faculty of Psychology and Neuroscience at Maastricht University, and she was chair of the reading committee.
Thank you, Pro-Rector. Dear candidates, first I would like to congratulate you on this thesis. It was very diverse. You didn't only work with animals, you also worked with humans, but you also didn't do just behavioral research, you also did EEG research, and you used pharmacology partly. So that is very impressive. But of course, I also have a question for you. And this relates to chapters 2 and 3. So in those chapters, you did 2 animal studies. And in the first, you looked into memory retrieval, but in the next one, you looked into memory consolidation. But then in both chapters, you also used different memory tasks, because first you looked into spatial pattern separation, and in the second one, it was object location. And I'm now wondering, because you say you showed results only of the first because they were the same in both studies, but can you really say that because of the fact that you had different types of tasks, but also because you were looking into different types of memory, consolidation versus retrieval? So how complementary are the findings really? I would like to hear that from you.
Highly esteemed opponent, thank you very much for the compliments for my work, and appreciate your question. So in chapter 2 and chapter 3, they are separate different tasks, as what you just said. In the chapter 2, this is basically about different part of hippocampus. This is mainly about to check the dental gyrus, this part of the hippocampus. And in chapter 3, that is mainly about the general hippocampus functions. So we separate the different chapters to focus on different function of the different part of brain to explain whether PDE4B or D inhibitor can work on different place. This is the idea of why we separate the chapters.
Okay, so you separated based on the location in the hippocampus that plays a role. But then about the memory retrieval versus memory consolidation, does this now mean that PDE4D inhibition has an influence on all memory because it is affecting different phases? Or what do you think about that?
So from the results we have, I think that could be the points. The first, in chapter 2, we sleep deprived before the pattern separation test. So that's we disrupt the retrieval, and we use the PDE4B and D. We found that they can improve that performance. So that could say these drugs can actually work on the retrieval process. And then on the chapter 3, we sleep deprived mass after the learning trial. And we can also see the similar results. So I think PDE4B or D can work on both memory process like consolidation and retrieval.
And maybe to continue on this, what if you had used object location in the first study with the memory retrieval? Would you have found the same thing? Do you think you would have found the same again? And also in chapter 3, if you had used the pattern separation task on memory consolidation, would you also have found the same thing?
That's a really interesting point. We could try to flip it, like do the different tasks in a different memory processing. From my perspective, I think that could be the same results, but we need to further data to support these ideas. That's, yeah.
Okay, and maybe finally on this one, for the study in chapter 3, where you say, okay, we looked into memory consolidation because you gave the drug after the learning trial. Could it also be due to a change in memory retrieval, that finding, rather than memory consolidation, or are you really sure that you have measured memory consolidation there?
I think this is mainly about memory consolidation, because normally consolidation happens after the learning. After the learning, then we let them sleep. During this period, they will have a memory consolidation process. So when we did this in our experiment, so we can know for sure that this processing is really about consolidation. And we give the drugs during the sleep deprivation to see whether they can prevent this deficits happen.
Okay, so the drug is out of the system when they do the retrieval. and therefore it can't be memory retrieval that is enhanced? Or would there still be sufficient of the drug available in the animal's brain when they are doing the object location task?
I'm sorry, would you mind repeat that?
So you give the drug after the learning trial, and then you make them sleep or not, and then they do the then they do the recognition trial. Is the drug out of the system when they do the recognition trial, or is there still some of the drug available in the system that could also influence memory retrieval rather than only consolidation?
Thank you very much for this question. We give the drugs during the sleep deprivation. And that's actually based on the half-life of the drug. So for the PDE4B inhibitor A33, that is mainly about 4 hours. And for the PDE4D inhibitor, that is zotomilast, is mainly about 10 hours. So based on this half-life, I think it mainly works on the consolidation. So I think that would be the point.
Okay. Thank you. Do I still have some time, Prorector? Yes. Then I have a question on proposition number 7. I don't know whether anyone would like to read it for us.
Sleep facilitates cognition by enhancing memory encoding for the coming day and consolidating newly acquired information from the previous day.
Thank you. My question is, because this is a general statement that you made in the propositions, did you prove part of this statement in your thesis? And if so, with which of the studies did you prove it? And for the part where you did not provide proof in this thesis, why do you think this is the case, what you've written here?
So in my thesis, I only proved the consolidation part. And for the encoding part, it mainly happens to before the learning trail. So if we want to know what happens to the encoding phase, we have to sleep deprived mice before the learning. We didn't do that in our thesis, but I think in the future that could be a good direction to explore further.
No. And yeah, so the memory consolidation you looked into for the animal studies, and what about the human studies that you did? Do you think you also found proof for the memory consolidation part of this proposition?
So thank you very much for this question. In the human studies, we actually gave the subliminal reactivation before the recognition phase, and we sleep deprived them after the learning phase, but not directly, but on the night of the learning. So I think that could be the point that we disrupt consolidation process. Yes, but we need further data to support that point. Yeah.
Of course. Thank you very much. I'd like to give the word back to the prorector.
Thank you very much. The opposition will now be continued by Professor De Hooge, Professor of Biological Psychology at University of Leuven in Belgium. And Professor De Hooge was also a member of the reading committee.
Thank you, Prorector. Esteemed candidate Zhao, I first of all would like to comment on your thesis. As my learned colleague also said, this is a remarkable piece of work. You've used many different techniques and you've studied animals as well as humans, so that covers already quite a range. Moreover, it also looks visually very nice. So it's, the layout is very beautiful. I would like to continue a little bit about these different kinds of memory, but a little bit at a different level. Because you've been using, in humans, the word learning task, the picture learning task. Some of these things are situated in what we would call declarative memory. Whereas in your animal studies, you looked at the object replacement test, etc. So my question is, since your title is memory deficits, and do you think you've been testing the same kinds of memory in your human volunteers as you did in your animals?
I think in humans, like you just said, we mostly did declarative memory, and we choose all the hippocampal-dependent memory tasks. So hippocampal-dependent memory tasks is mainly like a subset of the declarative memory. So it's and for the animal studies, we also use hippocampal-dependent memory, like spatial pattern separation or object location memory. So I think that's kind of similar because all of them are hippocampal dependent. It can be the subset of the declarative memory because during the encoding and the consolidation phase of the memory processing, it's mainly dependent on the hippocampus. Then in the later process, maybe the system consolidation is going to go to the neural cortex. That's maybe less related to the declarative, but less related to the hippocampus. But for our study, both human and animals are all related to the declarative, and that is hippocampal-dependent memory.
Mm-hmm. So would you be so bold in saying that the object location test is an animal model of declarative memory?
It's not a kind of model, but it's really sensitive to the sleep deprivation.
Okay, well, let's continue also with the same comparison about the sleep deprivation, because of course you're using very different methods in your animals to induce sleep deprivation than you did in the human volunteers. Do you think there's a comparison there? Do you think that the severity was the same in your animals as compared to your humans? And do you think if the severity would be different, would you also actually do something completely different in the two species?
Thank you very much for this question. So for the mass, we specifically do the sleep deprivation during, give the drugs during the sleep deprivation. But for the humans, we actually give the subliminal reactivation before the memory recognition. It's kind of like different systems because we cannot use drugs so far in the humans. But I think that could be the future direction. For the system, it can answer the same question. All of them, both animals and human study, can answer the same question. Like sleep deprivation-induced memory deficits can be recovered by both subliminal reactivation or drugs like what we used. And it can answer another question that the PDE4 inhibitors or non-invasive brain stimulation can reverse that deficit.
But would you agree that the severity of the sleep deprivation is different in your design in animals compared to humans? Because in your humans, you asked them to skip an entire night of sleep, didn't you? Whereas in the animal studies, it was only a couple of hours.
That's because mass and human have different sleep system. Mm-hmm. So for mass, they have really fast sleep cycle, but for humans, is have a longer sleep cycle. So that's the difference of the different duration for the sleep deprivation. It's fixed for the different species.
Yeah. That was exactly my point, actually. So that's why I was asking, do you think you've been doing the same thing?
I think so.
Yeah.
Yeah. I think so. We are doing the same thing because for example, in the humans, for example, in the mass, we sleep deprive them on the we use the sleep deprivation and the animal paradigm fits the circadian rhythm because mass is nocturnal animals. They usually sleep during the night and awake during the day. So we sleep deprived on the early phase of the light. So that means they already active for the whole night, for the whole day for them. Then we sleep deprive them. It's actually similar to the human to sleep deprive them at the night of the learning trail. Because they are different species, so we use different methods.
Mm-hmm. Okay, thank you. I have a question about the phosphodiesterase inhibitors as well. Isn't it counterintuitive that such a drug would improve memory? Because in your introduction, you've been explaining how memory works, and it's about, of course, storing information in the brain based on an information network of specific contacts. Whereas when you systemically apply PDA inhibitors, you would enhance the cellular function of neurons indiscriminately, all over your brain, all over your hippocampus. So isn't it strange that with such an aspecific cellular stimulation that you see an improvement of memory?
I'm sorry, would you mind repeat that question?
Yes.
Yeah.
I was wondering why a drug that indiscriminately enhances cellular function, like a PDE inhibitor. I mean, it inhibits the breakdown of second messengers, doesn't it? And still, it affects all these different neurons. But of course, in memory, it's about stimulating a specific network. So isn't it strange that this drug is still enhancing memory?
Well, yeah, actually, for sure, different subtypes work on a different brain area. For example, PDE4B inhibitor mainly related to the thalamus, hypothalamus, or striatums, these areas, and PDE4A or D maybe share the brain area like hippocampus or cerebellum, this area. And I think all of these inhibitors like A, B, and D can work together to affect the memory like hippocampal memory we used. And alternatively, they can work in the same cell, but distribute differently. It's okay, but all of them can work together to both contribute to optimal signaling transduction. So I think it's not a problem.
Yeah, but wouldn't you expect that this drug is just increasing noise since it's stimulating all neurons in your network, not only the ones that are storing the information?
I don't know for sure, because I think later we could use more data to explain that questions. Because from our perspective of the results, we cannot tell you anything about that use like definitive results.
Yeah, you could of course hypothesize that your drug is only enhancing the ones that are relevant. Could that be a mechanism?
That could be.
Okay. Thank you, Mr. Prorector.
Thank you very much. The opposition will now be continued by Dr. Meerlo, Associate Professor of the Neuroscience, in particular Biology of Sleep at the University of Groningen, and he will address his question online, and he was also a member of the reading committee.
Thank you very much, Rector. Dear candidate, like the others before me, I want to congratulate you and your supervisor with the completion of this wonderful thesis. What I really appreciate in the research of you and your supervisors is that the work is deeply rooted in basic neuroscience and builds on very recent findings. But then you are taking it to the next level, and you look for realistic, feasible, and direct applications in humans. And I think that's wonderful. There's not many labs who make that big step from basic science to application. I had a long list of questions, but I'm going to start with one that was not on my list. It was triggered by your introduction and by the discussion that you had with the previous opponents. And that is the question as to why it looks as if the hippocampus in particular is sensitive to sleep deprivation. And it looks as if memories that make use of the hippocampus are more sensitive to disruption by sleep deprivation than other kinds of memories. And I'd like you to follow up on the discussion that you already had and maybe enlighten me as to why you think that is the case. So why is the hippocampus more sensitive than other parts of the brain? And do you think that has something to do with the mechanisms that you are studying?
Esteemed opponent, thank you very much for your compliments for my works, and thank you very much for the questions. I think for the question about why the hippocampus is really sensitive to the sleep deprivation, I think that's mainly about the memory processing when we have sleep during the night. Because, for example, in the encoding phase, it's like when we sleep well before the learning, it's actually like reset the synapse, like making more room for new information to come in. And for the consolidation part, it's mainly like slow wave sleep, or maybe rapid eye movement sleep, or stage 2 of non-rapid eye movement sleep. They are really sensitive to the hippocampus memory, because they can do, they related to the declarative consolidation, or like procedure memory, or memory or emotion experience. Then for the last part is about retrieval. It's also sensitive to the hippocampus memory, because it's related to how people recall the information they learned. So sleep actually can emphasize this kind of process to give more energy for the people to recall the stuff.
Yes, thank you very much for this answer, but I'll still continue on this same point a little bit, because in your introduction you talked about these memory processes, and you showed a little bit of the potential molecular mechanisms, the involvement of cyclic AMP signaling. Now that's a hallmark of memory processes, but so therefore, I don't think it explains why memory processes that occur in the hippocampus would be more sensitive to sleep deprivation than memory processes that are associated to other brain regions. So what do you think is different in terms of molecular or cellular mechanisms when it comes to hippocampus memory versus other memory, like cortical memory or amygdala-associated memory? So what is different between these brain regions and the molecular processes that makes the sleep deprivation more effective in hippocampus memory? Do you think it could have something to do with the PDEs that you are studying? Are they differently expressed in different brain regions, maybe?
Thank you very much for your questions. So yes, for sure, we have different kind of memories, like declarative memory, like apositic memory, or semantic memory, or other memory like non-declarative memory, like priming, or classical conditional. This kind of memory, they are all actually can be affected by sleep deprivation, but in my thesis, I only focus on the hippocampus memory. So for the other part of memory, I think in the future that could be a good direction to follow up.
Yeah, but so you are now one of the world specialists on PDE, of course. I'm not, but are these different PDEs, and especially the ones that you were targeting with your drugs, are they differently expressed in different parts of the brain, and are they differently expressed in the hippocampus than, for example, in the amygdala? Do you know?
Mm-hmm. Yes, they are work on different part of the brain. For example, PD4B is more related to the thalamus or hypothalamus striatum, this area, and also PD4A or D is more related to the hippocampus or cerebellum, this area. They are works on a different brain area. Then I think maybe they can work on the different functions. But from my study, I cannot tell that much, because we only explore more about the hippocampus memory related to the like object perception or spatial memory.
Okay. Okay, so that's something interesting to follow up then I guess. I mean, throughout your thesis, you're emphasizing the importance of the hippocampus for memory processes and the role of sleep in this. And then somewhere in your introduction, you're saying slow-wave sleep supports system consolidation by reactivating and redistributing memories to sites of long-term storage, while subsequent REM sleep aids synaptic consolidation by stabilizing the redistributed memories at their long-term storage site. So what exactly do you think these long-term storage sites are? Where are they? Presumably outside of campus.
I think long-term memory. This can be happened following the CAM signaling pathway. It's like, for example, maybe everyone can look into the page 11. That's the mechanism of CAM signaling pathway. It's also related to long-term potential. Maybe we can begin with AMPA and NMDA receptors. They are all glutamine receptors, and they are both located on the postsynaptic membrane of hippocampal memory. AMPA receptors are the faster responders when the glutamine bonds. They quickly open and allow the sodium ion flow in. That causes depolarization of the neurons. And this can also activates NMDA receptors. When NMDA receptor activate, it can allow calcium ion flow in to activate the cascade like AC CAM signaling this pathway. Then after they activate PKA, phosphorylate of CRAB and phosphorylate of the GLU-A1, they can also increase the AMPA receptor insert into the brain. I think this part of the how that memory can be processed like the long-term potential.
But I think what you're saying in the introduction suggests that the initial memory formation starts in the hippocampus, but then with long-term storage, it is transferred to another part of the brain.
Transfer to
is that correct?
Yes, sure. Because first they learn something and store in the hippocampus, and in the long-term system they transfer to the new cortex to stay longer, like long-term storage.
Yeah. So and you attributed specific roles of slow-wave sleep to this initial hippocampal consolidation and in the transfer to another brain region and to REM sleep in stabilizing this in this long-term site. So on which part of this process do you think your treatments are acting? Is it the transfer or is it the long-term stabilization?
Can you please keep the answer short, due to time?
Okay, thank you. I don't think we focus on the long-term processing. We only focus on how memory started. That's on the hippocampus memory. But it's a good idea to following experiment to focus on the long-term storage.
Okay, thank you very much.
Thank you very much. The opposition will be continued by Dr. Augstulewicz, Assistant Professor of Cognitive Computational Neuroscience at Faculty of Psychology and Neuroscience at Maastricht University, and he was also a member of the Reading Committee.
Thank you, Prorector. Dear candidate, thank you for the presentation, very, very clear, and I would like to also congratulate you on this body of work, so congratulations to you and to your supervisors. I was, as other members of the committee, really impressed by the cross-species aspects of your approach and how many different methods you use to support your propositions. So one of the assumptions you take is that, or one of the things that you actually try to introduce is this concept that subliminal memory reactivation might be a human applicable alternative to optogenetics. So did you consider any alternatives to subliminal reactivation, for example, non-invasive brain stimulation? And if you did, why did you choose this particular topic for your research?
Esteemed opponent, thank you very much for your compliments for my work, and thank you very much for the questions. So for me, I think subliminal reactivation is optimal choice. The first is because it's actually mimic how optogenetic actually works. Optogenetic actually reactivate memory engram, and subliminal activation use the learned word in the learning phase. So it's actually the similar mechanism to reactivate memory engram. And also subliminal reactivation can relate it to the natural sensory processing. That means they can mimic how brain process the memory. Unless maybe other methods like TMS or TES, they actually activate the general cortical brain area. This can lead to interference, like unnecessary interference. And also subliminal reactivation is less expensive. We don't need to pay that much on the equipment, or we don't need to do some dangerous surgery for that. So I think for my perspective, subliminal reactivation is really a good choice. But for sure, we could try other non-invasive brain stimulation, like what I said, TES or TMS, or other approaches. That may be a good way to go.
Mm-hmm. And to show that this is a good analogy to optogenetics, don't you think that you would also need an animal study using subliminal reactivation? Or perhaps you know similar studies from the literature?
Yes, I think we need something like subliminal reactivation to work on the animal studies, because so for now we already know that sleep deprivation can be memory deficits caused by sleep deprivation can be retrieved, and we already know subliminal works in the humans. But what we don't know is the mechanism behind it, like whether subliminal activation can actually activate memory engram. So I think this is something we can do in the humans in the future. Then we can actually use intracranial EEG in the mass, that we can see what happens in the brain in the animals.
Mm-hmm. Thank you. So on this topic of subliminal reactivation also, this will be a rather technical question, but I think nevertheless important. I noticed that you use these very short presentation, of course, 16 milliseconds, 32 milliseconds in some studies, right? But your refresh rate of the monitor is also around 16 milliseconds. So it's actually the difference between 1 and 2 refresh rates. Did you actually measure it independently or validate it to make sure that you're presenting as short as you want to present?
Yes, our monitor is around 60 hertz. So that means each frame lasts around 16.667 milliseconds. So that is the shortest duration we choose at 60 milliseconds. But that doesn't matter, because the duration we choose is based on the previous study. They showed that subliminal prime always shown between the 10 millisecond to 50 millisecond, and we are in this range. And also we did another pilot, but actually it's in the chapter 4 studies to choose the optimal duration. And we found that 60 millisecond is around or below chance level performance. So I think that doesn't matter.
Okay, thank you. Now, you also measure EEG or ERPs, you calculate ERPs to show that there is this effect of subliminal reactivation. But you focus on very specific ERP components, and I just wonder why you went for this approach, because when I look at the plots, it seems that the difference between stimuli that were previously subliminally reactivated and those weren't, the difference is similar to some of the other parts of the ERP that you didn't analyze. So why did you go for these specific components?
Thank you very much for pointing this out. We choose the different ERPs. Actually, it's based on some of them is based on the previous study. We look for the subliminal cues and the recognition memory, and the ERPs we choose is mainly related to them. And then it's based on what we focus on our study. So, for example, in the subliminal phase, what we're looking for is some ERPs that shows maybe relate to attention or subliminal cues or early visual processing. So we choose P100, P200, and N200 as is related to these meanings. Then in the recognition memory phase, we mainly focus on the recollection or something like familiarity, this kind of stuff. So we choose the ERPs like LPC, like P100, N170 to explore more about this this focus.
But just to explore your data even more, wouldn't it be an idea to look at the entire time window so that you can actually discover potentially new signatures of previous reactivation?
Yes, we actually also did like visual inspection about all the general, about the ground average. We actually did more than this, but we only found some significant difference on the thesis that you can see. Yes, for sure, it's better to explore more like other ERPs, but we did that actually.
Mm-hmm. So your subliminal reactivation method, that actually is supposed to reactivate a memory engram, right? So it's about memory contents. But then the ERP analysis just looks at the amplitude differences. So it's whether activity is stronger or weaker after reactivation. Did you consider any types of analyses, for example, decoding, that you could actually say something more specific about the information in the EEG activity pattern?
That's a really good point. Yes, for sure. For the EEG we use now, it's not explain that much about mechanism. It's mainly tell us why and how that subliminal are processed in the brain. So in the future, we could use that methods to explore a bit more like the mechanism behind it.
Mm-hmm. And so one measure that you also included in the thesis, but maybe not so much in the presentation, was this perceptual awareness scale, right? But here you used it only at the very end of your stimulus presentation, and in the EEG study, actually, you didn't use that, right? So could you tell me why you went for only using it in this specific way?
Thank you very much for pointing this out. Yes, for sure. This perceptual awareness goal, we only use that in the chapter 4. We didn't use that in chapter 5 and 6. That's because chapter 5 and 6 is not focused on the optimal duration. For example, in chapter 5, we mainly focus on like to validate the subliminal reactivation, this method. In the subliminal duration, in the subliminal phase, we actually say the brain activities can, its difference depends on the different type of stimuli. And in the recognition phase, we find that brain activities is different depend on with and without subliminal reactivation. And in chapter 6, we mainly focus on the sleep deprivation. So this is now all of this chapter is not relevant to the perceptual awareness score. So we only use that in the chapter 4 because we want to use that as supplementary data to support this duration we can use in the following experiments like chapter 5 and 6.
Okay, thank you very much, and thank you, Prorector.
Thank you very much. And the opposition will be continued by Professor Drinkenburg, Professor of Translational Neuroscience of Psychopathology from University of Groningen.
Thank you, Mr. Prorector. Dear candidate, allow me to first thank Maastricht University and especially your promoter, Professor Blokland, for the opportunity to read through your very interesting thesis. And I can only echo what was said by earlier opponents, that it's an impressive body of experimental work, and I read it with a lot of pleasure and interest. But I also found that in the last chapter, you made some interesting statements which I would like to have your further thoughts upon, especially with respect to the translational and therapeutic impact of your work. You state that the studies that you've done offer a translational perspective, highlighting its potential to inform future therapeutic approaches for mitigating cognitive impairments associated with sleep deprivation. Now, I can follow that. That is more or less a direct outcome of some of your interesting findings. But in the next paragraph, you continue, and you say that these non-selective PDE4 inhibitors also show promise as potential treatments for neurodegenerative diseases like Alzheimer's disease. And you state that the overlap in underlying mechanisms between the memory deficits caused by the sleep deprivation and Alzheimer's disease underscore the translational relevance of your findings. Now, when I read that, I was a bit lost. I wasn't quite sure whether I properly understood, and let me tell you why. I do understand that both conditions impair hippocampus-dependent memory functions, and they do share disruptions in underlying pathways. But as far as I know, in Alzheimer's disease, the chronic short sleep is there for a much longer time. And the short sleep has been associated, for example, with glymphatic clearance system that's very active during sleep. And the consequence of that is that there is a lot of neuroinflammation, issues with the microglia system. But that may well be a completely different mechanism than what you have seen after your acute sleep deprivation. So my question to you is that, is the time course of the effects in your studies and what's underlying Alzheimer's disease, are they not completely different? And can you really extrapolate from your acute findings, acute sleep deprivation findings, to the chronic, more neurodevelopmental and neurodegenerative diseases like Alzheimer? Now, I hope you can educate me there and make me rethink my reasoning.
Esteemed opponent, thank you very much for pointing this out. It's a really good question. So sleep deprivation actually is rare, happens in isolation. It's normally co-occur with other disease like neurological or psychiatric disease like Alzheimer's disease, depression. For example, in the early stage of Alzheimer's disease, the sleep structure already changed, so it can lead to memory decline. And also some of the disorders that you just mentioned, they can be cured. They can be treated by other drugs or other administration. So that means the memory on these disorders, they are not fully lost, but hard to retrieve. So that is similar to what we found in my thesis. And I think this disease and sleep deprivation, they share the similar brain area like prefrontal cortex and hippocampus, this area. So we could hypothesize that they may share the similar signaling pathway. And actually one paper already shown that Alzheimer's disease and sleep deprivation, they share the similar pathway, that is CAM signaling pathway. This is what we found in my thesis. So I hope that I answered your question.
Yeah, I obviously good research always raises much more questions. So I have another question then for you. The silencing of the engrams, to me, is always a very interesting phenomena, and there are several reasons why that can happen. And it may well be that what happens during Alzheimer is comparable to what you are inducing here. But how do you extrapolate that to schizophrenia? Because schizophrenia, you mentioned that as well as a possible therapeutic application, at least of some of the work. I think that has a slightly different problem in terms of cognitive disturbance. Is that not less memory retrieval dependent issues in schizophrenia? So what's the nature of the cognitive deficits in the two disorders?
I'm not sure for what's the difference between this disease, but I think what we can do, the application from our thesis, we could use subliminal reactivation, this method, to check whether Alzheimer's disease, their memory are completely gone or still there. So we can use subliminal reactivation to let patients to do following experiments. Then first give them some learning trail, then give subliminal reactivation. If they can recall, that means these memories not completely gone. So we could use drugs like PDE4 inhibitors or like other non-invasive brain stimulations. And also, I think non-selective PDE4 inhibitor like Hofmilast, they already passed phase 2 clinical trial. That mean they can use the early stage of the Alzheimer's disease. That is what I can explain here.
Okay. And maybe as a final question, do your results on your evoked potentials, your ERPs, they seem to point towards, I mean, you use them to show the activation, but do they also show towards the nature of the difference in information processing? So I got the impression that it was mainly the very early, the rather early components, not very early, but the rather early components, which usually are more related to attention than to cognition. So would that be something that you would then more relate to schizophrenia rather than to, for example, Alzheimer's? How do you position your ERP outcomes with respect to the two different diseases?
Because we only found the different ERPs in sleep deprivation studies, so we can only explain that in our result. I mean, early ERP studies can. So maybe
you
we
can finish your sentence.
Okay.
Yeah.
So the early ERPs that explain the attention and visual process. Maybe we can continue the conversation in the reception.
I would like that. Thank you very much.
Hongyu Zhao, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know side. No place like home. If it's questions that you've got, go the extra mile or die. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Did they say 25 or did they say 1?
Hongyu Zhao, the committee has decided to award you the degree of doctor at the Neuroscience, Psychology and Neuroscience faculty. The chair, Arjan Blokland, Professor Blokland, will now award you the degree.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Hongyu Zhao, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I will present you the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Congratulations.
Now comes the most difficult part for me. Ja Hongyo Bosch. Chongjin Jun Ni. I hope you understand it.
Brilliant.
I won't try it again. I just want to say, dear Dr. Chang, Dr. Hongyu, congratulations with your doctoral degree. It has been an interesting journey with you, because we first, I will repeat the Chinese later on again, maybe. Yeah, so we had contact with, it's a very complex process that Pim and I went through, because we had to submit, we have to meet you, and then you have to program, and then this, and then that, and eventually we got the message, okay, Hongyu Zhao can come to Maastricht. And then we had a wonderful plan what you could do. And actually, when I now look at the plan that originally I looked to my previous mails, when I looked at all these studies that we had in mind, there's only one study that we actually did from the original plan. So we had to move here and there to the other things. And I think we really triggered you to do all kind of different things. So we first said, okay, we will do some animal studies, and we gave them some drugs, and you didn't have any experience with drugs, psychopharmacology, these kind of things. And you had to go through the animal course to learn all these things. And then we had to learn how to sleep deprive the animals. And actually, we also dissected the brain and at the frontal cortex and the hippocampus. And actually, once I was sitting there, I had to keep the mice awake because nobody else was in a situation to help. And I saw you with such a precision and such a speed of dissecting the brain that was just amazing. And just learning that in a really short period of time. So you really mastered that wonderfully. And we were just, and when the data came in, they were just as we would expect with the sleep deprivation. So it was really, and you also experience what you need to experience when you do animal experiments. You're bitten once by a mouse. And then you know what it is to do in animal experiments. But then, because of different reasons, we had to switch to human studies, because we thought, of course, that would be much more interesting. So we started to think about, okay, what could be an interesting way to replace this optogenetic study, optogenetic methods that Pim was using in Groningen. And yeah, we were moving to humans, so, well, we came up with the idea of subliminal stimulation, having no clue whether it will work. So we were very much out in the dark. And then we had to present subliminal stimuli. And I think one of the opponents already mentioned that, so the 60 milliseconds, that was really a problem. Because first we learned you to program in E prime, and then we looked at the screen and we saw two peaks. So actually the subliminal stimuli were presented twice. And then we had to switch to presentation, another program, and then we could exactly see that the 60 milliseconds just presented at 60 milliseconds. So it was really a kind of you forgot to say that, actually, but we spent quite some time on that and finding the right program to do that, yeah. So that's also, you learned E prime programming, presentation programming, and then we had to go to this sleep deprivation study. And then you managed to find all these participants, and they had to do quite some interesting and very not so exciting studies, because they were looking at a screen with subliminal stimuli, but they didn't see anything. And say, come on, you have to remember. So these participants say, well, what do I need to see? Yeah. So it's quite tough to get these participants through these studies. So you really should be commended for great work you did. Yeah. And then we thought, well, no, that's not enough. We also need EEG. So then we said, well, Haley, you have to work on EEG. So learn how to put the cap on the head, make the electrodes work well, and do the test, and then do the analysis. So that was also quite an endeavor. That was quite an interesting period for you, I think. Yeah. So you did all these things, and with no complaints. You said, yes, sir. And then you did all these things. And actually, it culminated in a really interesting thesis, and I think we need to do a little bit more with the human study, but this really interesting data, and Pim is looking forward to publish that in a very high journal if we do the other things. So, but that, from this experimental things that you all did, was just great, fantastic. Yeah, and you were also, I still remember that you were really angry with us when we were talking about your experiments and you didn't know about it. And you said, this is my experiment, I want to know everything about it. So we were really a bit, okay, now we inform you, we didn't keep you out of anything anymore. Yeah. So, I mean, that was good. You stand for what you were doing.
Thank you.
Well, yeah, okay. Yeah, so also the communication, it was really fast. We had mails coming back after sending it. It was just immediate reply, and always ending with "Have a nice day." And that was so. You always had wonderful days when you ended your email. Yeah. The writing was more tough, and we had some. By the way, you look beautiful in your dress, I must say. Yeah, and your hair is now really. But sometimes your hair was really wild. When we had the discussions about the paper, how to write it, and then she was really into it, and she was looking like a wild Haley. Yeah. But that was the writing phase. That was very stressful, I think. Yeah. And of course, you needed to finish the thesis within a specific time because, you will start soon your work again in China. Yeah. So when we first met you, your English was quite good, and we were quite happy with the proficiency of your English. But as soon as you came to Maastricht, you made all kind of Chinese friends, which a lot of them are here. And we noted that the English was actually not getting better being in Maastricht. It was actually... And I saw the laptop. First was all these folders with all these English names. But suddenly when we sat together behind you, I was only Chinese characters everywhere. And then this laptop changed in a Chinese laptop. Yeah. So, and I hope we can still find out what is where and what we can find back on that laptop. As mentioned, you have a lot of Chinese friends here, so, and you were really social. You invite people to your apartment and have hot pot. So we once had the hot pot at Haley's apartment, and I think she invited 100 people, and we had to eat it with like 10 people or so. Yeah. So we were bulging, like our bellies were bulging like crazy. I had to recover like 2 days from that hot pot. Yeah. It was really nice, and a really nice invitation. And you were open to many people and do things together. You actually were also traveling. You have seen more of Europe than I did.
No.
Yeah. You went to all the places in Europe with a friend of you. Maybe it was you? Or?
Yes.
Yeah. But you also picked some wrong timings. Sometimes you once went to the Keukenhof, the famous Keukenhof, but when you came there, it was closed. Yeah, you should be, or you went to Texel, a place that normal Dutch people don't go because it's windy, rainy, and it's nothing like a tourist place. Yeah. So you should have asked us for better places in Holland. Yeah. You also went back to China because you had to apply for your position at a medical school. And you were studying for the exams, you had interviews, and I think you stayed there for about 3 months. And in between the exams and the interviews, you were making progress in writing. So you were, although you were in China, you were still doing all these things. And yeah. And you ended up eventually with finding a suitable, it's not your primary choice, but it's Shanghai that you were going to. But at least you did, I think Shanghai is a really good medical school. And so you should be happy with yourself that you ended up there. And when she came back, and also on other occasions, Haley never forgets to give a present. And now I forgot the flute. I wanted to show you how the flute works, but yeah, unfortunately. But I got the flute, I got a cup of tea, I got mixtures of tea, really interesting. Already by looking at the tea and the leaves in there, and there are more things in there than just leaves. Yeah, so I've tried one, and it was an interesting experience, the Chinese tea. Yeah. And I also I received from Haley the book, The Free Body Problem, which I had to read, of course. And then when I was ill, I had my back issues, I got the other 2 books. So now I know everything about free body problems and how in the future our world will be conquered by other people. Yeah. So I hope you will have a wonderful future and in your next phase of being trained as a psychiatrist. And I hope you will find your work atmosphere and China back again. So maybe your English will get better. So, and we already talked about maybe coming to Shanghai one day, and we'll see. I also would like to thank the opponents, especially from outside, for taking the time to be here and doing all the work. And with that, once again, congratulations, and I give the word back to the rector.
Thank you very much. Dr. Zhao, I would also like to congratulate you with your title. Also on behalf of Maastricht University and the Faculty of Psychology and Neuroscience. Now, before I close the meeting, I have a few practical announcements to make, and this concerns the taking of photographs. First, the committee and you collect here to make a picture with the online opponent in the background. The second picture will be on your special request, so you can direct it, but I understood it is you in front of the audience. And the third one, and only after that, please, the public can move to the reception. So please stay primarily for a while. And the last picture, we go to the stairs with the committee again. Okay? So Having said that, I hereby conclude the session.